cone snails. Cone snails are really, really beautiful, and they wash up on the beach. And most of these cone snails you could find in the South Pacific. And so a tourist walking along the beach in the Philippines sees a beautiful shell, and they pick it up. Well, the animal inside of the shell is still alive. And so it actually has a harpoon at the for this is shaped like a cone, and at the narrow end of the shell is where the harpoon sits, and they shoot the harpoon out in a defense mechanism, and that injects their venoms into the unsuspecting tourists. Well, the tourist initially feels intense pain from the area of the injection, followed by muscle weakness, and this muscle weakness will progress. And eventually, if they get enough venom, the person cannot stand any longer and has difficult breathing. Um, and so, if this person does not get medical attention soon, they will die because of asphyxiation. Their muscles won't work and they can't breathe, and so they'll die of asphyxiation. So this cone snail has a lot of different components in the venom, and the venom, the venom, these components will target various different types of things, parts of the vertebrate muscle system. What these cone snails are doing normally when they're not washing up on the beach is that many of them are hunting fish. And so what they want to do is, if you look in this thing, they're shooting out their harpoon to harpoon a fish, and this little cone snail doesn't want to be dragged around the bottom of the ocean by a fish, and so what they're trying to do is paralyze the fish as rapidly as possible. And so they have a lot of different components to their venom that are going to target various areas of the neuromuscular system. So there's a lot of these cone snails. Many, many, many different species of them, and you can see they all have really beautiful shells, and so people will be tempted to pick them up. So if you see one, don't. So there's somewhere in the order of estimated, it's not known exactly, 500 to 700 different species of cone snails. Each of these species, their venoms have over 100 different peptides. And so Dr. Oliveira at the University of Utah, our collaborator on this project, calculated that if you do the math, it's 100,000 some odd pharmacologically active peptides in that these various different species have. So it's a lot of potential perhaps beneficial drugs that are out there. So there's so many of these different components to the cone snail venom. They've been characterized molecularly, and so you've got the different families are named by Greek letters. So there's alpha, and alpha are nicotinic acetylcholine receptors. And so the nicotinic acetylcholine receptors are the component that triggers the muscle, the action potential in the skeletal muscle. So by blocking that, the cone snail can paralyze an individual. There's another group of peptides in this venom that are called mu for muscle. So the original voltage-dependent sodium channel toxin that was isolated here was a muscle targeting the muscle, and so again, the action potential in the muscle being blocked paralyzes. And then the one we're going to focus on today are the omega conotoxins. And so omega conotoxins target voltage-dependent calcium channels. Now, the promise of this that was seen back in when this research was started at the University of Utah by Doctors Oliveira and Gray, they saw this huge potential and said, well, let's start breaking down, then looking at these various venoms and the various components of the venoms to see is there anything that can be medically useful that we can get from these toxins. And so to that, we have to start talking about calcium channels. Voltage-dependent calcium channels are broken down into nine. There are nine different genes that make these channels. The CAV1 group, the CAV2 group, and the CAV3 group. So we're going to talk about the CAV1 group later, but right now I'd like to focus on, just as a background and to bring home the potential medical benefits of looking at these cone snail venoms. I want to talk about the calcium channel CAV2.2. Now, this calcium channel is really, really important for pain signaling. And if you can block this channel, you can provide analgesia. And there is one component of one cone snail venom that was developed beginning with Dr. Gray and Dr. Oliveira's work, and then further worked on by pharmaceutical companies, developed into the drug zaconotide. And so these are a number of different cone species. And so all of these species target these calcium channels, the CAV2 group. And so this is just an example of the breadth of how important the cone snails think this target is. So a lot of them have these venoms, but one of them, Conus magus, called M7A, was developed into the drug zaconotide. And so on the human CAV2.2 channel, it has a nanomolar affinity for that channel. So it's very, very, very potent. And so what it's doing is, here we have a primary nociceptor, and primary nociceptors in pain, lots of action potentials, and those action potentials come into the dorsal horn of the spinal cord, and they trigger that depolarization triggers these voltage-dependent calcium channels here in orange, and these calcium channels release the neurotransmitter glutamate onto the second-order nociceptors, depolarizing. That causes lots of action potentials, and when these action potentials reach the brain, the person that has these action potentials is going to perceive pain. What zaconotide does is it very selectively blocks this particular type of calcium channel. That's the little purple thing here. And so by blocking that, there's fewer calcium channels that are available. And with that fewer calcium channels available, less of this depolarizing, this neurotransmitter glutamate is, and so there's less glutamate here. There's a less depolarization of that second order nociceptor. And there may be fewer or no action potentials that are generated by this second order nociceptor. And if there's fewer or zero action potentials, there's less or no pain, because action potentials are the high frequency action potentials are the signal for pain. We block the action potentials, we block the pain. So this was worked out into a drug. And so this is some data from Elan Pharmaceuticals, which eventually developed the drug into zaconotide. And so you can see here's an animal flinch test, and the higher numbers here means pain. And so 2 days after giving the injection of either zaconotide or morphine, you can see, well, morphine provided some analgesia, but zaconotide did as well. 7 days, morphine's back to normal. Zaconotide is still blocking the pain. So with one dose of zaconotide, you can get long-term pain control. And so for patients that have failed oral analgesic medications, this medication is an option, but you have to go to a pain management specialist to get it, because they need to insert a catheter into your spinal cord to deliver the pain directly into the vertebral column in order to deliver it and block the pain. So what this does, it gives us an example of the power of this, of looking at this. So we've seen that using these cone snail venoms, we can develop useful medications. And so we wanted to further look at this and say, well, okay, we found peptides within some of these cone snail venoms that can differentiate between the CAV2 group. Can we find them that would differentiate between the CAV1 group? And so the CAV1 group, the two that we wanted to try and differentiate was the CAV 1.2 and CAV 1.3. Now, where are these located? Well, unfortunately, they're located in many of the same tissues, in the central nervous system, in the heart, in endocrine cells. And so there are a series of drugs called calcium channel blockers. And these drugs that are currently available as medications do not differentiate between these two channels. And so they'll block one and they'll block the other. Now, that could be good, but what it could possibly also do is increase the potential adverse effects. And so what we really like to have in our medication is something that is very selectively targeting one component or one particular channel in this case. So the question is, can we find while these drugs that we have here on the side and on the right-hand column, they're medically available. There are no known drugs that will select between these two different channels. And so that was the motivation for this study. And so a little bit more about these channels. What do they do? Well, in the heart, they're found in the AV and the sinoatrial node, the atrioventricular node, doing pacemaker, generating action potentials. In the cardiomyocytes, they're in the ventricular and atrial myocytes, helping to to generate the contraction, but also working with excitability, particularly in the atria for CAV 1.3. They're in the brain, and so they have various functions within the brain. One of which is in the substantia nigra, where they're regulating dopamine release and so that shows in the next slide that there may be some a role that the CAV 1.3 in particular plays in Parkinson's disease. Also in the adrenal medulla, releasing adrenaline, spontaneous activity, and aldosterone secretion, the hormone aldosterone. And then 1.3 is really, really important for our hearing and in the vestibular system for our balance. And it's actually generating the calcium signals that are critical for us to be able to hear and balance. And so they have some important roles to play. Now, too little of it. So if we have too little of CAV 1.3, we have bradycardia, slow heart rate, and cardiac arrhythmias. If there's too much, we can have Parkinson's disease. If there's too little in the ear components, we can lose hearing. And if there's too much in the adrenal cortex, we can have hyperaldosteronism and treatment-resistant hypertension. So if we could have some selective medications that would either enhance the activity or block the activity, perhaps we can develop them into drugs that could help with these various diseases where increased or decreased activity of CAV 1.3 is important. All right, so inhibiting CAV 1.3 activity, treatment-resistant hypertension, Parkinson's disease. I didn't mention seizures. There's evidence for a role in seizures and depression. Enhancing the activity, potential treatment for hearing loss. We mentioned cardiac bradycardia and cardiac arrhythmias. So either enhancing or inhibiting you might be able to develop some beneficial drugs. So that was our motivation. We're going to look through these cone snail venoms to try to find selective blockers or enhancers of either CAV 1.2 or 1.3. And so the way that this was done, and all of this work was done by my colleague here at KCOM, Dr. Kim Han. All of the data that you're going to see, except for one piece of data, was generated by her. The other piece of data that I'll show was generated by our colleagues at the University of Utah. And so what we did is we got these clones of human CAV 1.2 or human CAV 1.3, and we transfected them into a cell line, and the cell line is called human embryonic kidney cells, and they're very, very good at expressing foreign proteins, and so they also have few or none ion channels of their own, so they don't interfere with our signals. And so they're a great system to be able to look in isolation at these various components. We could look in neurons, but neurons have many different ion channels. They have many different calcium channels. They have many different potassium channels. And as we know, the reason we're looking at this is we don't have any selective blockers for CAV 1.2 or 1.3. So what we want is a system where we have only CAV 1.2 or only CAV 1.3, and then we can look at to see, does this component block this or that or both or enhance? What do we see? So what we're showing you here is so there's a DS red is a indicator that's showing that the cells were transfected, and then what we've done is we have labeled the cells with an antibody to on the left side here, or my left, I don't know if it's your left as well, CAV 1.2 transfected cells, and this antibody is in the upper row is selective for CAV 1.2. In the lower row, the antibody is selective for CAV 1.3. And you can see that we have a nice labeling of the CAV 1.2 antibody of only the cells expressing CAV 1.2. On the right side, it's the reverse. We've transfected the cells with CAV 1.3, and again, the same antibodies, but only the cells expressing CAV 1.3 are labeled by the CAV 1.3 antibody, basically demonstrating that we've got selective expression of the channels that we want, and there's no other components, significant other channels that we have to worry about. All right, so what's the experiment? So we've taken our cells and we've transfected them with either the calcium channel 1.2 or calcium channel 1.3, along with one thing I forgot to mention in the previous slide is that there's three different components, protein components of this channel. There's what's called the alpha subunit, and that has all the gating mechanisms, and the pore allows the calcium to get into the cell. And then there's a couple of modulating proteins, the beta receptor, and we're using beta 2A, and then there's another modulating receptor, alpha 2 delta. And so the alpha 2 delta is interesting because it is a target of a drug called gabapentin or pregabalin, and those are very important analgesic medications targeting this particular component. But we've put them all together because we want, as much as possible, a real channel. and a strong channel that works like it's normally supposed to. And again, these are human channels, and so which is again what we wanted. So we have our transfected cells. We put them, we load them with a calcium indicator. So these are calcium channels, and we put them into a solution that's going to have calcium on the outside. The calcium, as it comes into the cell, will create a fluorescent signal because of this fluorescent indicator. And so this is a calcium indicator. The higher the calcium concentration inside the cell, the brighter the fluorescent signal is. Then we measure this using a Leica confocal microscope, and that takes images every 5 seconds. Now, these are voltage-dependent calcium channels, and so what we do is we depolarize the cells with high potassium. And so, as you can see here, whenever the high potassium pulse goes on, we get an increase in the calcium signal, which goes back down to baseline, and then we do it again, we do it again. We do 3 of these to make sure we have a stable signal, and then apply the venom. The venom goes on, the venom or the fraction of the venom or the peptide isolated from the venom goes on for 10 minutes, and then we wash it off. And if there, in this case, there's no effect because the potassium signal, you can see the calcium signal is identical throughout. At the end, we put on 100 millimolar potassium just to give a maximum signal. So this is the type of experiment that's going to be done for every one of the venoms and fractions of venoms that we are going to use. By the way, all these images are analyzed with image J to generate these time course graphs that we see here. And so just to look at, here's our control experiment. And so in the upper panel, this is control. So control is the solution that we're putting these venoms into a solution. And so we want to make sure the solution itself does not cause a change in our calcium signal. The upper traces here are from CAV 1.2, the lower traces are 1.3. Throughout this presentation, any bars that are in blue are from CAV 1.2. Any bars that are in orange over here are in for 1.3. And so as you can see in this example, there's no effect of the control solution. And here are the over on the right side here, the data from many cells showing that. Now, we have a drug called nifedipine, and nifedipine is a drug that can be, it's FDA approved. It can be used in humans as treatment for hypertension, for example. And so it's a blocker of both of these channels. And so what we're going to do is we're going to put 10 micromolar nifedipine on and then see, does a known blocker block our signal? So this was our control. The above is our control. Does our control solution have any effect? This lower one panel is another control. Does a known blocker have the effect that we expect? And as you can see, both the CAV 1.2 and 1.3 signals are very strongly blocked by nifedipine. And here are the group data off to the side, to the right side, showing that. So the system works, and so which is good. And now we can get to the data. So we were sent from the Oliveira lab 11 venoms from these 11 cone snails. And so we went through and did these. And so here are the data. And so the most promising data that we got came from Conus geographus. And Conus geographus has generated other very selective calcium channel blockers in the past. And so that was interesting. Another one that was not significantly different, but it was based on data that came to us from the University of Utah, our collaborators. They said Conus Gloria Maris also was of interest. And then later on, we found that another cone snail. Again, these are not significantly different, but we were getting a signal, and then the data that we got, or information we got from our collaborators at the University of Utah said this was another, Conus radiatus was another um, venom of interest for them. So then what happens if we have a venom of interest? Well, so if we look at Conus geographus first, so this is what our collaborators at the University of Utah do is they take these venoms and they inject it to a high pressure liquid chromatography machine, and then they separate out as a first pass the components. And so this is an example of a component map from a venom. And then what they'll do is if we have a venom now, Conus geographus, that they're interested in, they'll take fractions of this and they'll isolate these fractions. They'll capture these fractions, and then they'll send us the fraction and say, can we identify a fraction that has a similar profile or an interesting profile similar to what we saw from the entire venom? And so this is an example of fraction 1 from Conus geographus. And so here's the fraction applied to the CAV 1.2 and 1.3. And so what you can see here is that this fraction inhibited 1.2, but enhanced 1.3. So that was interesting. So we have a significant enhancement of one component while inhibiting the other. Now, these fractions have many, many components still. And so what we were thinking is maybe we can find a very potent inhibitor of CAV 1.2 in this, selective, and maybe a potent enhancer in this. All right, so here's the data from fraction 1 that I just showed you. The fraction 3, that was a bunch of inhibitors, and then the other fractions we tested just inhibited 1.2. So let's look at fraction 1. Now that's been subfractionated, and subfraction 1, fraction 1, subfraction 1 gave us a similar effect as the fraction itself. So now we able to find this subfraction that seems to be of interest. Unfortunately, we didn't get any components of that subfraction. Our collaborators at the University of Utah did not want to follow this up. What they wanted to do was follow up fraction 3. And in our data, fraction 3 didn't give any significant differences. In fact, both channels were inhibited. They had some different information, and so they wanted to fractionate subfraction 3. So now we have 3 subfractions from that. And we found one that had a significant inhibition of CAV 1.3, while a much less inhibition of 1.2. So that was encouraging. And I think this follows what their data were supporting. So fraction 11, or subfraction 11, was then further differentiated out into individual peptides. And so we have a number of different peptides, and all of these things were tested. And basically, we didn't really find anything of particular interest. This was the original subfraction 11 that we were hoping to find something that was very potent for 1.3 block. There was one component, peptide 9, that gave us a moderate, maybe a 37% inhibition of 1.3 and no real inhibition of 1.2, but it wasn't significant, and we didn't find any significant difference of any other component. And so for some reason, we weren't able to reproduce this effect of the subfraction. And so for that reason, while this may be of interest, this was dropped because it wasn't significantly different. All right, so this is our sort of our sheet. We've got, this was a very initially a very promising component, and out of fraction, the pool one, we have again promising. Um, that ended up in something that they did not want to follow up on. And since they were supplying us with all the venoms and fractions and subfractions, that's if they don't want to supply it, we don't get it. Um, Peptide 3, on the other hand, or sorry, fraction 3 was further fractionated, nicely done there. We had something, but then when we looked at the individual peptides, nothing came up. And so, unfortunately, this is a common theme in this talk. So Gloria Maris was something that they thought was interesting. We found one peptide that significantly inhibited 1.3 more than 1.2, but then when that fraction was further subfractionated, we didn't get anything out of that either. So um, that one that line of work ended. So that left us with radiatus. Conus radiatus was, so just to remind you, since it was a while ago since we looked at this, Conus radiatus had, this is not a significant difference, but it was a strong enough difference. And what they had, they actually had from their data a significant difference here. And so they wanted to look at this, at Conus radiatus venom in more detail. So they sent us a series of fractions. We had 11 fractions from this. And basically, nothing was particularly of interest except for fraction 9. Fraction 9 gave us a significant inhibition, a stronger inhibition of 1.3 versus 1.2. So that was interesting. And we got, again, for the third time, hope that we could isolate a component that would selectively inhibit one or the other of these components. So fraction 9 was subfractionated. And so subfraction 32 gave us some interesting data. So this is the percent block. And so subfraction 32 at 30 micromolar gave us a significant block of 1.3 versus 1.2. That was exciting because now we've been able to follow that from the original component down into one of the subfractions. And so here's the data here, 53% block of a reduction in the CAV 1.3 signal, only a 17% block in the CAV2. Now, as pharmacologists, we like to do dose responses because dose responses gives us a better idea of what's going on. So we started doing both dose responses. And unfortunately, when we did the dose response, well, as you might expect, the 1.3 signal got further blocked, but so did the 1.2 signal. And now the signals are no longer significantly different. So what this begin to look like is that, yeah, there's maybe something here that's going to be somewhat selective, but it's not that selective. And so we did not further examine subfraction 32. What we did look at, and again, based on our data of a very strong block of 1.2 signal without, if anything, an enhancement of 1.3 signal, very strongly significant effect. They wanted to investigate this, and so they fractionated this in the subfraction 29 was further fractionated into individual peptides. And so we had two peptides of interest. Peptide 12 showed a significant inhibition, and peptide 14 showed a significant inhibition. And they were very interested in looking at peptide 14. And so here's the data from peptide 14, showing 1.3 here on top, no effect of this, while as 1.2 calcium signal was inhibited. We looked further using a dose response relationship here, 3 micromolar of peptide 14, 10 micromolar, 30 micromolar. At CAV 1.3, no effect, which was really encouraging, and increasing inhibition here with at 1.2. So again, we're really seeing what we think could be a robust effect. So here's what our data looked like. We went through 12 cone snail venoms, 28 fractions, 17 subfractions, and more than 22 peptides to find peptide, Conus radiatus peptide 14. And they were also looking at this, and these data come from our collaborators at the University of Utah, and what they looked at were mouse dorsroot ganglion neurons. And so what they found, and so what they do is they dissociate these dorsroot ganglion neurons and then depolarize them and then find out, then they put on the blocker and does anything block. Now these these neurons are from many, many different subtypes. They're all neurons, but these different neurons will have different levels of calcium of the CaV2 calcium current. And so what they were looking for is the block by 10 micromolar of this peptide 14. And so they found many cells that showed really no block, a very mild block, a moderate block by some cells, and then a couple of cells showed a very strong block. And this gave us some, and so we've actually looked at the calcium currents in these neurons, and this actually matches with the populations of cells where you would expect a strong component of from the CaV 1.2 channel. It's actually, there's a minority of cells that show a very strong component of 1.2. So this actually was encouraging, and it actually fits other data that we have. Just looking at the component calcium channels in these dorsroot ganglia cells, neurons. All right, so we were very encouraged. And so the other technique we have to look at this is using electrophysiology. And so here is a calcium current. These are voltage dependent CaV 1.2 currents. And so we make a depolarization, we record, and this is a inward current, the current that we want. And then we will do a time course. So we repeat this over and over and over again. And so we see a nice stable recording. And then we put on BSA. BSA is bovine serum albumin, and it's a carrier for our peptide. Sometimes these peptides will stick to the tubing that we have that we're using to apply these substances to the cell, and it's very common in venom work to use these carriers, and so we've used it. And so here we put it on, and nothing happened. And so we washed it off, and nothing happened. Just to check, we put on our nifedipine, and we got a very nice block by nifedipine, exactly what we expected. So the venoms that we tested and all the peptides were on for 10 minutes when we do our calcium imaging. And this is only applied for 2 minutes, but we've also applied it for 10 minutes. We've applied it from the outside, we've applied it from the inside, and results are all the same, disappointingly negative. We have not been able to reproduce the calcium imaging data with electrophysiology, and that is a problem. And so for that reason, we're hesitant to publish this, because we don't want to put out something in the literature that's not reproducible. And so basically, we've identified components using calcium imaging of from the cone snail venoms that looked promising. From the Conus geographus venom, fraction 3, subfraction 11, peptide 9, looked like a promising blocker of CAV 1.3, but it wasn't pursued. Conus radiatus venom, fraction 9 had a couple of different components. Fraction 32 looked really good, but while it wasn't both components were blocked, but our collaborators didn't want to pursue that. What they wanted to pursue was a different fraction, and so subfraction 29 was differentiated into peptides. We found one that looked very promising in the calcium imaging. However, we have not been able to reproduce this using electrophysiology, despite multiple application durations, techniques, various things. I presented this in Austria and everyone was really excited. And I told them we haven't been able to reproduce it using electrophysiology. Got some really good ideas, suggestions from our colleagues there, brought them back. None of them worked. So we are really, really at a frustrating standpoint right now where we don't know what our next step is going to be. However, if we could find some blockers that were selective for either 1.2 or 1.3, that would really help us understand the physiological roles of these channels. Because as you remember from the physiology, they are co-expressed in a lot of different tissues, and trying to dissociate out what their exact role is somewhat challenging. And so having some selective blockers would be wonderful. There could be also perhaps benefits for treatment of diseases resulting from the aberrant channel activity, but that remains to be seen. And with that, I will take any questions you might have. Sir.
I'm just curious, as it's a really neat system and all. The smallest subfractions you have may still have multiple compounds in it, or do you think you're down to a single peptide?
Well, we are with that peptide 14, we are down to a single peptide that they've actually synthesized.
That's what yeah. And so you can synthesize them, and then you can get a bunch of it in a more pure form if you wanted to, for more consistency in your tests, I guess.
Exactly. And what they do is once they get it down to, they sequence it, and then they've got multiple synthesizers, peptide synthesizers, and then they will synthesize it and make a lot of it. And then if it's some peptides are more challenging to synthesize than others. But yes, it's this has now been synthesized. And so
And
some
if,
of the last work we were doing was with the synthesized peptide.
and if the snail were modifying the peptide, you'd see that, because I'm assuming you're sequencing with mass spec, so you'd see if there were modifications, phosphates or something like that on them.
Yeah, I'm not aware that the snails will modify will phosphorylate or something. These venoms, basically the components sit in the venom sac, and if they are modified, it's possible that the something could take, if it was really important that phosphorylation was critical to the potency, and then there was something around in the venom sac that was going to dephosphorylate a phosphatase or something like that, that could be a problem. I think they're not modified that way. I know the component, the ziconotide, which is the drug that's used to treat pain, is not modified. It has no post translational modifications on it. But yeah, those certainly, it's possible that there are modified, but I don't think these were. they've also they actually started um, mutating the sequence in order to try and get it to be more potent. But that hasn't really gone anywhere. And since we can't verify the original peptide, it's, modifying it later on is probably not doing further modifications isn't, probably may not be the most useful thing until we've got it verified.
No, I wouldn't even suggest, but I just was wondering if that, could explain. And boy, but it sounds like with so many sequences, it's like you must just be scratching the surface of this whole thing, right? There's so many to explore and stuff. And that's why I guess you're doing these batch studies and stuff to try and zoom in.
Right. Yeah, there's thousands of peptides, or well, at least hundreds of peptides in each venom. And it's possible that if we were to go back and some of the venoms that didn't look very interesting as they got subfractionated, maybe something would come up. But you basically follow your best signal and then um, take your chances. Unfortunately, we so far haven't been able to identify something that was robust in using different techniques. Yeah, so there's no other questions, then oh yes, a question.
I want to ask a question. Why do you think the effect goes away when you further subdivide the fraction?
So there's a couple of ideas I have, and I don't know. We've not talked with them to see what their idea is, but if you've got multiple components within the venom that are sort of doing a little bit of, say, inhibiting a channel a little bit, and you combine them, now you've got a lot of inhibition. But then as you separate them back out again, you see that there's no one component that is that's providing a dominant effect. What our hope was is if we had say a 50% block of a venom that had a 50% block of CAV 1.3, our hope was that there is a peptide in that venom that is extremely potent at blocking that channel. And so the idea is as you fractionate and isolate, the blocking should get better, not worse. Because now instead of we're adding an amount of venom that has a small amount of each one of these different peptides, and then as we fractionate it, we add that same amount. The concentration of those peptides within that fraction are now higher because it's the same amount as it's now in the fraction instead of the whole venom. And then as we focus further and further down to the individual peptide, we would see potency go up and up and up and up. And that's not what we've seen. So
Okay.
it's probably a mass action effect. Everybody's contributing a little bit.
But then maybe it's still worth analyzing all the different peptides from this one fraction that has the inhibitory effect, and then maybe look at combinations of those.
Yeah.
Or is that going to be too much? I don't know what
I
numbers
think
we're
when you
talking.
start looking at combinations, so
Yeah.
it's already been, I think, we've been doing this for 6 years. And so that's just trying to look at individual components. And Dr. Kim Han was getting pretty tired of uh, basically doing things, finding interesting things, and then hitting a wall and said, "Oh, and so she said, okay, I think we're done with this." And so unless we can find another avenue for, a way, another idea on how we can get this thing to work in using different measurement techniques,
Mhm.
then we're putting it up on the shelf for now.
But they know a lot of those different peptides. I mean, couldn't you do something like a just like a computer programming, kind of seeing what components are maybe binding to the channel or something to kind
That
of
would
reduce their
require a channel a channel structure that was really well known.
so
And
it's not known.
no, the problem with transmembrane proteins is they don't crystallize very well. And so that's the standard techniques to get crystal structures from these has been, and so what's been done is there's some bacterial channels that have been crystallized. And so as soon as those channels were those structures were published, everybody took the sequence. Well, all the channels have the sequences, the amino acid sequences are known. And so they took the sequences and they mapped them onto the structure and they made structures, but who knows if they're real or not. They're based on a bacterial structure. There's some new techniques, atomic force microscopy, they're beginning to get the resolution down into the nanometer range where you could actually begin to get structures. And so they're trying to do that, and that's progressing. But right now, that's to my knowledge, there's not a structure. And then you also have to fold these peptides properly. And so exactly how these peptides fold, not being a peptide biochemist, actually a biochemist at all, then I'm really way out of my comfort zone here. But maybe there's something, but then so our collaborators at the University of Utah are biochemists, and so if these things are available, then they would be doing them.
Mhm.
How many amino acids roughly? This is like below 100 amino acids in these, or I mean, on average.
Yeah, so the zaconotide peptide that is that's approved for human use has 26 amino acids. The peptide, this 2914 peptide, I think 10 amino acids in it. Could have been it could be a little less or a little more. I don't remember exactly. Um, and so these are um, generally small peptides. Um, Now, the interesting thing about that zaconotide, it's got 6 cysteines in it. And so it's very, very tightly packed because you get um, the uh, um, the disulfide bridges. You get 3 disulfide bridges formed in these peptides, and so they're very, very they're very stable because of that. They're hard to digest because the thing is so tightly wound. The only way to digest them is basically easiest way is to break the disulfide bonds. But so this particular peptide, I don't think had any cysteines in it. So it's how it works or how it folds up again is not something that we did not talk to them about that to see if they've looked at it.
Well, you don't
But
know the sequence of the ones you're working with right now, or
yes, we do.
oh, and you say no cysteine in them, or you don't think there are.
I don't remember any cysteines in it.
Yeah, because that's kind of weird to think, because normally you want to keep everything in a highly reduced environment to avoid whatever, but with those, you might need to keep them in an oxidized environment so that the cysteines don't come apart and relink in different ways or something,
Yeah,
because
with the ones that do disulfide bond, the cysteines are those disulfide bonds are very stable. And so you could keep those peptides. I mean, we've used them for various experiments, and that you could keep them out on the bench overnight, and they're just as potent the next day. So It's quite good.
I'd be afraid to put them in a reducing environment. Yeah, I'd rather them on the bench.
Yep. Yeah, you don't want to you don't want to separate them out because they're or break those bonds. But this one, if it had a cysteine, I don't think it had any cysteines in it. So all right, well it's approaching 1, so or well 2 o'clock here, 1 o'clock there. So it's been great. So thank you for the great questions. Nice conversation after the presentation. So.
Thank you, Dr. Elmslie. And if you have any other questions, please feel free to email us and we will send those questions to Dr. Elmslie.